Hi, welcome back to Hydroponics Daily with me, Dr. Russell Sharp. Yesterday's topic was polyploidy, and I said that today we'd be talking about micropropagation, and we will. I just want to revisit polyploidy for a second because I missed a couple of key bits out. The first one is, how do you confirm your plant is polyploidy? Maybe it's just a mutant. And what you have to do is you take a microscope slide of some of the cells of your plant, and you stain them with a chemical called safranin. And this is a chemical that's extracted from saffron, as you would imagine, because of the name. It's the red dye in saffron. However, you can just buy the pigment. You don't need to extract it. And then you just stain the cells, and then the chromosomes, the DNA takes it up, and then you can count them really easily in the nucleus of the cells. And that will confirm whether they have artificially increased the chromosome count, or if they're back at what the wild type plant should be. You might also want to do this to confirm the chromosome numbers, so if you're looking to cross them and things like this. However, that's fairly advanced, and so is taking microscope slides. It's not something you just whack under a microscope and do fairly easily, so you need to be pretty good at using a microscope to use those sorts of techniques. The technique that I used, this was my first job ever in plant science, was counting chromosome numbers on microscope slides, and that was at Ness Botanical Gardens on the Wirral Peninsula. It's part of Liverpool University, and my tutor there was Professor Hugh McAllister. Shout out to Hugh McAllister and all the other plant scientists and hydroponic growers in Liverpool. Liverpool is a hotbed of hydroponics, so I'm sure we've got some listeners on Merseyside. I'm from Merseyside originally, but unlike Steve and Barry from NPK Hydroponics podcast, I don't have a Scouse accent. And that's because I went to university to study plant science and nobody could understand my accent, so I had to change my accent. All my family speak with Scouse accents, especially my sister. But yeah, I'm a dad. But I don't speak with a Scouse accent, but I am from Liverpool originally. And yeah, so that's polyploidy. We'll move on to micropropagation now. It's a technique linked to polyploidy because you can induce polyploidy very easily if you apply the chemical colchicine to the calyces, the undifferentiated growth, the blobs of growth that you get in micropropagation. However, that's not the only reason you would want to micropropagate a plant. There are loads of reasons you might want to micropropagate a plant. And that's normally because either you're struggling to propagate them from cuttings or grafting, or you're having little success in germinating seeds, or it takes a long time using conventional methods. These are all reasons you might want to micropropagate a plant. Another term for micropropagation is tissue culture. So if you're struggling to find resources on micropropagation, you'll also find stuff on tissue cultures, or microprop for short. Okay, so in its simplest form, it's producing new plants from small amounts of tissues, excised from the mother plant under sterile conditions and cultured on a gel medium. And by far the most important word there is sterile, because it's a bit like brewing. In micropropagation, you spend 99% of your time cleaning things and making sure that everything is sterile. Same in brewing. Brewing is just cleaning stuff. And it's a really good technique. It's very scientific. And there are a few amateur micropropagation kits now available. Athena started selling them. They're quite expensive. I think they were about £1,000. And there's a big hype around them. Not sure if anyone used them successfully to micropropagate any plants when they were released a couple of years ago. And you might think, the Athena, they're making loads of money out of that. But it's a fairly complicated kit. The materials are not cheap, and getting them all together couldn't have been easy. So, fair play to Athena for putting it together. No one else was making micropropagation kits. And I would imagine that was probably a loss leader just to make sure that they were seen as the sort of leaders in scientific growth in hydroponics. So, kudos to them for trying. They're a competitor, but at least they did something different. And then, micropropagation. So you may have heard of micropropagation in your industry. It might be some niche users, or it might be standard. I mean, if you're cultivating orchids, carnivorous plants, ferns and palms, bamboos, then micropropagation is the standard way they are propagated. There is a company called Micropropagation Services in Nottinghamshire, and they micropropagate sphagnum moss, and they then put them into little beads of agar, And they get into a helicopter and they sort of like fire them out of a helicopter like it's Vietnam in the 1960s and 70s. And they pepper these moss balls all over peat bogs and moorland to reintroduce sphagnum moss, which is a key structural species in these environments. Really interesting one. Another example of where it's been used to propagate rare plants is the yellow lady slipper orchid, which is, in the 1990s, I think it went down to about one plant in the wild. And then Kew Gardens, the biggest botanical gardens in the world, used micropropagation to raise the plants of this rare orchid. But it's used on a lot of Asian orchid nurseries and things like that. It's the standard way. I've seen micropropagation used on bamboo and orchid nurseries in Holland and many times. And this is an amazing fact. If you have a micropropagation lab of 23 square meters, which is not big, that is a standard bedroom, you can produce up to 1 million plants annually. So that is crazy numbers. And with an initial outlay, you can do this for under £2,000 or $3,000. But, these setups can run up to hundreds of thousands, even millions of pounds. When you start putting in the grow on and the greenhouses and the grow rooms to actually get those into bigger plants. One of the words you'll hear often in micropropagation is weaning. And just like a baby, weaning them off milk, you have to wean them off the agar and the sterile environment. That's what the specialist greenhouses are for, is that you need to wean them slowly in very high humidity and then lower the humidity as the stomata become adapted to this and the growth forms. You can't just keep them forever in 100% humidity. Before I go into the details of how you actually micropropagate, there's some other good uses for micropropagation is the eradication of viruses from infected stock plants. So imagine you've got a strain, a variety that is really popular in your industry, but the mother plants get infected with a disease. And then you can use micropropagation to remove the virus. It's a very good way of removing viruses. And so things like hops, things that get riddled with viruses, that would be a good option. So we said weaning is a word that's used often. Another word that's used in micropropagation is one of my favorite words in science, and that is totipotent. And totipotent means that basically a meristematic tissue, a plant from the growing bud, the meristem of the plant, can turn into any of the cells programmed in the plant's genome. So that undifferentiated cell could turn into, if it's from a carrot plant, it could turn into a carrot root, it could turn into a root hair, it could turn into a xylem vessel, it could turn into a phloem vessel, it could turn into a leaf stomata. Sorry, not a stomata is just the whole. A guard cell could turn into a petal, it could turn into a pollen tube, it could turn into anything. And that was discovered by a British botanist, Frederick Stewart, working at Cornell University in New York in the 1950s. And he showed that you could produce a complete carrot plant from just a single cell. So that is 40 years before the first animal was ever cloned. That was Dolly the sheep, produced from a single mammary gland cell up in Scotland. So plant scientists have been doing this for years before the human scientists get involved. Same with virology. Plant science has been talking about their virus studies are way in advance of human viruses and animal virology for a long time. So it doesn't matter where the cells come from in the plant, unlike animal cells where there's sort of like a fixed destiny on the cells. Having said that, meristem cells are really the ones you want to produce new plants from. So when you're taking plants for micropropagation, when you're using a sterile scalpel, the first thing you do is you remove the meristematic tissue from the flower buds, probably an active growing one. You remove the leaves primordia, these are the young leaves around the stem, and then you need a microscope to dissect it, and then you put it onto a gel on a petri dish or in a tube with agar gel or some other sort of gel nutrient stock in there, and then just lay it on top and keep it in that, and seal it, keep it all sterile. And everything has to be sterile. The media has to be sterile. And so you use techniques like using IPA, isopropyl alcohol. You have an autoclave, which is basically like a pressure cooker where you make sure everything's sterile by heating it up under pressure. And then the first grow media is normally just a standard growing media with some nutrients in and some food for what they call the explants, these little blobs that you've taken from the merostem. And they need a bit of sugars in there because they're not going to be photosynthesizing at all. So they're going to be growing just like an animal. They're going to be feeding like a fungus or something like that. And they're just going to be multiplying by mitosis, and they're not going to be differentiating. And then eventually the clump will grow and grow and grow, and it'll form a callus, just like a callus on your foot or your hand, differentiated, a bit like a tumor. It's undifferentiated. And you just want to build that callus up. You don't want it to be turned into a plant too soon, because it needs to have energy reserves so that when it does produce roots and shoots, that it's got the energy to do so and maintain it. So as I say, the media contains all sorts of things like amino acids as well to make proteins. The most famous one is called Murashige and Skoog, and that's named after the famous plant scientist Toshio Murashige and Volker Skoog. They are the Lennon and McCartney of micropropagation. Together, Murashige and Skoog published the earliest definition of the technique in the early 1960s and developed grow media to culture the tissue on, and that grow media is still used to this day. And the recipes have been tweaked, but they're largely unaltered, and a lot of people still use Murashige and Skoog. It's known as MSO in micropropagation labs all over the world to this day. So the first one that you put the explant on will be high in auxins, and just like auxins in plants and rooting and rooting hormone, auxin will stimulate rooting. Before you stimulate rooting, though, you might want to cut the callus up again, especially if you haven't got much of the starting material to play with. So that example of the yellow slipper orchid. You obviously can't be going back and back to the one plant to rip it up to take cultures. So once you've got a big callus, you might then use your sterile scalpel to subculture and make more calluses and then build them up and keep those calluses going. But then you have stimulated the roots to form with the hormones, the auxin, then it's time to transfer them to a new gel medium with cytokinin hormones, and they stimulate the production of shoot tissue. And then after that, you've got everything you need for a plant. You don't need to stimulate flowers and fruits, obviously in a test tube or a petri dish. And interesting story about petri dishes. I'll tell you, I used to work at a place called Malton College, part of the Northampton University, and there was a student there who was called Petri, and Petri, and I said, "Oh, you named Petri after petri dish." And she got really upset, and Mum phoned in and said, "This is disgraceful, how the teacher was bullying her." And I said, "That's absolute nonsense. You should have seen it as a compliment, named after a famous scientist, Petri, who invented the petri dish and was a famous microbiologist." And yeah, got in a bit of trouble for that one. But yeah, F her in the A, to be honest. You're going to be called Petri. You've got to be expected to be called a petri dish every now and again. Where do you store all these plants all these calluses, these petri dishes and large test tubes? They're not really a test tube, they're more like a boiling tube with cotton in the top that allows the air to flow out and in, but prevents dust and bits of bacteria from getting in. You probably want to put them in a controlled environment. It's a bit like a grow room, but much more cleaner, no growing media around, no stagnant water. So maybe and then you can get almost like fridges with a little grow light in there. They don't tend to use LEDs or sodiums, certainly not. It's more fluorescent lights in micropropagation is fairly standard. And you just put them in there until they grow to the point where they start to outgrow the boiling tube or the test tube. Photosynthesis will be very low because the plants are in 100% humidity. They haven't got much water. And so there isn't much CO2 really as well. So growth is very, very limited and slow in micropropagation. So it can take up to 2 to 4 months to form a plant in micropropagation. But the good thing about micropropagation is you can keep them in sort of stasis as well. And so you could use them as a sort of safety net for your mother plants and as a sort of, I want to say repository. I always get confused between repository and suppository. As a repository for all your different strains and varieties, things like this. Whereas if you were storing them as fully grown plants, would obviously take up a lot of room. So imagine like a seed bank, but for living plants. So you use the autoclave to sterilize all the kit that's going to be in close contact with the plants. You would also sterilize all the cotton wool that you're putting into the boiling tubes. The grow media would be autoclaved as well, but then the things like the hormones, you don't want to autoclave them because they would break down. The amino acids would break down maybe under high temperatures. So you could filter sterilize them, which is another pain and another cost, but is required. The actual X-plant, you would sterilize in either ethanol or dilute bleach or something like this, probably ethanol. But that is going to be the source of most contamination, because that's where, it's going to be much harder to sterilize. And if the plant has got a disease or has got bacteria living in it, endophytes, they're called, they might not be harmful, but they might be living there, then they can be a source of infection. So that's really this can be the source where most infections going to come from in micropropagation. Often you use something called a laminar flow hood. It's almost like the opposite of a fume cupboard if you've been to a laboratory. This one pushes sterile air through at you as you sat next to the cupboard, and it's all filtered. And so all this air comes at you rather than being pulled up away from you like you would in a normal fume cupboard to take hazardous fumes in a chemical laboratory. But there's all this air flowing towards you. You've got ethanol, and you might have a flame to sterilize your scalpel and stuff like that. So it's a bit of a fire hazard. Once I was working with a colleague called Yannick down at East Malling in Kent, and he spilled his ethanol and it all went on fire. And he looked at Adam next to him and said, "Oh, Adam, I've got a problem." It's like, "Yeah, you have. You better come get a fire extinguisher." And he was on fire in this micropropagation lab where they were growing trees for shell, something like that, for one of their alternatives to petrochemicals. Poplars they were growing, that was it, yeah. So it can also be used to increase the sort of active ingredient levels of plants. There's interest in, sort of like from the pharmaceutical industry, using tissue culture to synthesize important drugs. So examples like the Madagascar periwinkle that's used for leukemia and Hodgkin's disease. But, currently it requires tons of plant tissue to obtain just 2 grams of active ingredient. So if you could massively increase the active ingredient content, that would be useful. And certainly other plants where you've got medical use, then they could potentially be used for micropropagation there, for not just for propagation, but for increasing content of the plants. It's important to know, just like cuttings, you always want to take the X plants from juvenile plants, not mature plants that have flowered, because it's always going to be harder to propagate from plants that have flowered. As soon as they've become mature, they're a lot harder to propagate asexually. It's just something goes on in the plant. I'm not quite sure what, but routinely it's going to be an issue. And as with all sorts of forms of asexual propagation, the plants produced by micropropagation are clones of the mother plant. However, mutations are common when they're in the callus stage. So, and polyploidy, as we talked about in the previous episode and at the start of this one, can occur in polyploidy, and especially if you stress them, if you put chemicals on like colchicine, UV light, X-rays, gamma rays, things like this, can all be used to treat plants in callus stages, and you can get mutations. Most and 99% of all mutations are deleterious. However, 1% at the time, you can get a nice, good one in plants. And it's not like mutating humans. There's no ethical issues because they're not going to feel pain or, have issues long term. It's just a plant. One notable success of micropropagation has been the rare Tunbridge filmy fern, which was thought to be or close to extinction in the town of Tunbridge Wells in Kent, and was extinct in Tunbridge Wells until 1875, and then micropropagation has been able to increase the numbers dramatically. Ferns are difficult to propagate because they don't produce seeds and flowers, they produce spores, and they have a haploid phase of their life cycle where they grow as a haploid for a bit, and so a single set of their genes. So really difficult to propagate ferns unless it's sort of asexually. So the Tunbridge filmy fern was a big success story. Oh yeah, I didn't mention when a mutation or a growth form that's new. It's often not a genetic mutation, just a growth condition, like growth deformity occurs in plants. It's called, in micropropagation, it's called somoclonal variation. And they pop up every now and again in micropropagation, especially in the large micropropagation laboratories. And they have been the main source of new varieties of hostas and heucheras in gardens over the last 30 years. Heucheras especially. Hostas have always been popular gardens, especially in the UK where it's so wet. Hostas really thrive. But heucheras are fairly, I'd say in the last 20, 30 years, really, really popular. And so the new varieties are often coming from the Asian micropropagators of these plants and all the different colours and growth forms. So lots, like almost like a rainbow of different heucheras now because of this. 3 more things to consider. I've been a bit back and forward, in and out on this episode. It's not really gone in a linear form, so please bear with me. If you want to eliminate viruses using micropropagation, which is probably what most listeners will want to do, I don't think many people are looking to breed or mutate. If you do want to do virus treatment, you want to heat, you might want to flash heat that callus to maybe 50, 60 degrees Celsius. And that could almost destroy the virus but not kill the plant. That's what you're looking for with virus treatments. And same with seeds. The seedlings, often if you flash heat them really quickly, then that can kill the virus and not kill the plant. When it comes to the weaning, you might want to include a mycorrhizal inoculant because remember they're going from a sterile environment back to an unsterile one, and you can't just rely on them, the grow media, having the optimum fungus in there. So that'd be a good one. And then even if your plant does propagate from cuttings, if it's really, really slow, then micropropagation could be an option as well. So the sort of house plants like the large leaf figs, they can be better micropropagated because they just take so long to propagate normally. You know, other plants that would struggle, will take a long time, like any sort of woody shrub like rhododendron or something like that, can take ages, even if you're really good at propagating them. So that's been Hydroponics Daily for today. Micropropagation, or tissue culture, is also routinely known. Please also check out our amazing fertilizers and crop inputs that we manufacture. So in America, that's gold leaf fertilizer. In the UK, it's liquid gold. Not to be confused with liquid gold leaf, which was a product that I used to make. I invented that formulation, but I've produced a better formulation now, and that's liquid gold. And you'll find that on the Utrema website, along with all our other amazing products that we sell in all different sectors. So we sell products to help protect your plants from pest and disease. We sell inputs for your lawns. We sell hydroponic fertilizers and biostimulants like seaweed extracts and humic acids. So if you think I've been talking some sense and you like the sound of where we're going with Utrema, then please do check out utrema.co.uk. Utrema is the scientific name for wasabi. So if you're struggling to spell Utrema, Eutrema. If you do go on there and you place an order, let me know you're a podcast listener and you might get a little treat in your package as well. Not promising anything, but let me know. I'm very, very forgetful, but just make sure you message me, send me an email, say I'm a podcast listener and I've placed an order, and I'll sort you out. Thank you very much for listening. I've been Russell Sharp. This has been Hydroponics Daily, and you've been an amazing hydroponic grower. Thank you very much. See you tomorrow.